Thank you, Paige, and thank you, John, for the invitation to be here. It's really been a delightful meeting and fun to spend time in Dublin. So what I want to talk to you about is work that we've been doing to try to understand the metabolic mechanisms that go into fatty liver disease. In particular, we're interested in a condition known as non-alcoholic fatty liver disease. You are probably familiar with it. It's one of the more common effects of obesity. In fact, in Dallas, where I'm from, about 30% of the individuals walking around in Dallas have this condition known as non-alcoholic fatty liver disease. So that's lipid accumulation that occurs in the liver somewhere above 3 to 5%. And generally speaking, it's benign. Most people don't even realize that they have this condition. It's not until kind of the chronic situation, chronic obesity, chronic insulin resistance, that they develop a more severe form of the disease called non-alcoholic steatohepatitis. And this involves inflammation and fibrosis. It can go on to lead to liver failure and require liver transplant. So we're really interested in what are the metabolic mechanisms that go into moving a liver from a benign condition to really a much more severe condition. So we actually know a lot about the metabolism of the liver during insulin resistance. As you're probably aware, the liver is producing more glucose. It's more active in that respect. In studies that I'm going to talk about today, we find that it actually increases substrate oxidation. In fact, there's data from the old literature that shows that in obese subjects, there's an increase in oxygen consumption. But over time, the liver really progresses to a state of oxidative stress and inflammation, and eventually to mitochondrial dysfunction that seems to be associated with this state. So what we're really interested in then is, are there metabolic mechanisms, are there metabolic pathways that might contribute to the progression to this condition and then this condition? And so that's what I'm going to talk about today. So I'm going to talk about some of the metabolomic and flux approaches that we use just in general, so you have an idea of how these experiments are done. And then I'll talk about the pathways in the liver, particularly mitochondrial pathways that might contribute to this progression. And then maybe I'll talk about some of the insulin signaling pathways that might contribute to changes in fluxes, and show you some experiments that really, I think, at least convinced us that these fluxes actually contribute to the progression to inflammation. So I want to start out just by giving you an introduction to the liver mitochondria and what makes liver mitochondria different from muscle mitochondria that we've heard a little bit about today. So this is just kind of the typical cartoon of a mitochondria. It's what we've seen in our first two talks. The mitochondria oxidizes substrates and generates ATP for work. In the muscle, that would be exercise or movement. In the liver, actually, it's chemical work. And so that's one thing that I'll come back to. The other difference in the liver mitochondria is that once you go through beta oxidation and generate acetyl-CoA, you're really not committed to oxidize that acetyl-CoA. In fact, during fasting, most of the carbons from your fat actually are diverted to ketones. So when you undergo fatty acid oxidation in the liver, you don't have to use it in the TCA cycle. In fact, you don't. You use most of it to make ketones. The other important difference in liver mitochondria is that the TCA cycle is not all about oxidation. In fact, there are these anaporotic and cataporotic pathways that shuttle molecules into and out of the TCA cycle, mainly for the purposes of biosynthesis. So if you're interested in what the liver does as work, this is what the liver does as work. This is how it spends its ATP, not by contracting. And so this is an important outcome that we try to pay attention to in the liver, because many of these things, notably gluconeogenesis, are elevated during insulin resistance and diabetes. So we'll talk about these pathways, and we'll talk about this pathway as we move through the talk. So So just to give you an introduction in how we study these things, I mean, we're interested in metabolic mechanisms, so we pay attention to methods like metabolomics and flux approaches. And I stole this slide from my colleague back at UT Southwestern, Ralph DeBaradinas. And Ralph likes to describe the difference between metabolomics and metabolic flux as counting cars. Counting cars in a parking lot. So if you're counting the number of molecules, any kind of certain molecule, then that's metabolomics. It can give you an idea of what the pathways are doing. But if you run a flux experiment, you're really interested in the movement of those cars through the pathway, as illustrated by this picture of cars traveling down the street. And I want to give you a real-life example of how these two pieces of data can give you different pieces of information. So this is from 2 mouse models that we have in the lab. It's real data. It's metabolomic data. We're counting cars. So we can tell that when we knock out one pathway or knock out the other pathway, metabolites are going up or going down, or they're going up. So just based on kind of the classical usage of metabolomics, what one might predict is that in this mouse line in blue, what you have is an upregulation of the TCA cycle, an increase in all the intermediates, right? And in the other mouse line, what you'd have is a decrease in the TCA cycle, a decrease in all of the intermediates. That's classically how we would interpret this data. But when we run a flux experiment, this is what we find. Both mouse models have a decrease in flux through this pathway. And the reason, I mean, that presents something of a conundrum if you're trying to interpret the data not knowing what the mouse is. But we know what the mouse is. And the reason this happens is because one mouse model, the blue bar, is a knockout of Pepsi-K, a cataplerotic pathway. The other mouse model, the gray bars, is a knockout of pyruvate carboxylase, an anaplerotic pathway. So really what we've demonstrated here is that when you knock out the entrance into the parking lot, there's less cars. When you knock out the exit to the parking lot, there's more cars. So a combination of the data can really be helpful in getting us to understand what's going on in terms of the metabolism. So this is a flux approach that we use in our lab. We're interested in the liver and in the TCA cycle. The liver will, if you give it labeled propionate or labeled lactate, it will take it up and it will convert it through this cataplerotic and anaplerotic pathways into glucose. And so we can measure the enrichment, the red dots, in order to measure this activity of anaplerosis and cataplerosis. But it's even better than that, because when the label enters into the TCA cycle, it gets diluted by acetyl-CoA, and so we can measure then the relative activity of the TCA cycle compared to these entrance and exit pathways. And we do that using nuclear magnetic resonance. And the reason we use nuclear magnetic resonance, so this is data from a carbon-13 NMR spectrum, is because each one of these peaks tells us about the population of these isotopomers that we call them. And then that in turn tells us about the activity of the pathways that we're trying to measure. And so we're able to go back then to the energetics and really study what we want to study. But we're a little bit obsessive about making sure these things measure what we think they measure. In fact, the field in general in flux is obsessive about these things. And so we want to make sure a couple of things are working properly. We know that this is a true tracer, because only 3% of the carbons are enriched, so we're not changing the pathways by mass action or anything like that. We want to know that our tracer doesn't actually induce any of the pathways that we're interested in measuring in. So we've measured endogenous glucose production in the presence and an absence of our tracer, and it doesn't change anything. We want to make sure that the internal pathways that we're interested in, the TCA cycle, anaplerosis, cataplerosis, none of those are changed. So we've done experiments with the tracer and without the tracer using an independent tracer, and this is the populations, they don't change. And then finally, we want to make sure that the pathway we're mainly interested in, oxidative flux, isn't affected by the tracer that we choose, and it's not. So just to explain, why do we pick this tracer? Why do we pick this method? It's because we can do it in both people and in mice in the exact same way. using the exact same infusion methods, the exact same analysis. So this is truly a translational method. We can study people, and we can study mice, and we can compare the mechanisms. So the final thing you want to know is, okay, that's all great, it doesn't change anything, but does it actually measure anything? So you want to be able to validate the method in that sense. And we can do that because it's not the only pathway that we measure. Using a variety of other tracers that I won't talk about, we measure overlapping pathways as well. And we can then put it into a more well-controlled experiment like the isolated perfused liver, where we can measure things that are coming in and going out. We don't have to depend on tracer approaches, and apply that to a mass flux analysis. That's essentially a fancy way of saying we're going to keep track of the stoichiometry. We're going to measure flux of the pathways. We're going to add up all the NADH, and we can measure oxygen consumption. And when we measure oxygen consumption and we calculate oxygen consumption from the fluxes, you get a very nice correlation. It's telling you that the method is measuring what you think it's measuring. That's very important to do with these flux approaches, because otherwise it's a black box. And so we try to do that. So these pathways that we measure using the tracer, the TCA cycle, oxidation in the TCA cycle, also ketogenesis, they're nutritionally responsive. So this is what the old literature tells you if you really dig into the liver literature about what happens to oxidative metabolism when you feed and when you fast. This is what's supposed to happen. Carbon is shunted towards ketone production in the fasted state, and it's oxidized in the fed state. And the liver's mainly a fat oxidizer anyways. If you have unless you're under a glucose load, it's mainly a fat oxidizer. But this is what happens if you put a mouse on a high-fat diet. If you put a mouse on a high-fat diet, you lose that flexibility. In the fed state, it's a little bit lower, and in the fasted state, it's a little bit higher. I'll show you that it's more pronounced as you go farther into a high-fat diet. So if you leave the mice on a high-fat diet, then you actually get an increase in this oxidative metabolism through the TCA cycle. Now, we did this experiment because we were interested in some of the fundamental mechanisms that our first two speakers talked about. Why does lipid accumulate in the liver? Does it accumulate in the liver because there's an impairment in fat oxidation? But we found out that there is no impairment in fat oxidation, that it's actually increased in this mouse model in the fasted state. But what really captured our attention at this point was that it seemed to be related to the amount of oxidative stress that occurs in a mouse liver when you feed them a high-fat diet. And of course, this leads us to the topic of this session, right? Mechanisms by which the mitochondria might contribute to disease in the fatty liver. Mitochondrial dysfunction, reductive load, lobular hypoxia. These are all ways that oxidative metabolism might contribute to oxidative stress. And this is what I want to try to test. So this is also true in humans. This is the data that I hinted towards at the beginning. This is an experiment from John Warren where they put catheters across the splanchnic bed in humans. And obese humans have an increase in oxygen uptake. We found the exact same thing in people using the tracer approaches, and these people also got biopsies. So the higher the oxidative metabolism, the worse their biopsy score was. The more along that progression line they tended to be. So it's really important that we understand why that occurs. And so what are the factors that regulate hepatic oxidative metabolism in the liver? Well, we've known for a long time that the more fat you give a liver, the more it will oxidize. Those are classic liver perfusion studies from the 70s. So that's important. But we wanted to know about the signaling, the molecular mechanisms by which that might occur. How insulin action and kinases that might affect downstream nuclear receptors to regulate that. But we were also aware that this oxidative metabolism is really important for the synthetic pathways that might be overactive during fatty liver disease or insulin resistance, and that could potentially feed back to the mitochondria to draw more energy out, to draw more ATP out. And then finally, it doesn't have to be a direct effect. The signaling pathways don't have to directly affect the oxidative pathways. They could do it indirectly by acting on some of these downstream pathways that use the ATP. And so these are the things that we're keeping in mind as we try to study this disease. And so insulin signaling is a mess. This is a vast oversimplification. It's really a rat's nest of pathways. But I want to focus on two potential pathways, because this is something called the insulin signaling paradox that I'll talk about in a few minutes. But when insulin acts on its receptor, what it does is it excludes FOXO1 from the nucleus, and then it decreases gluconeogenesis and all the downstream pathways. It also can act on another kinase called mTOR to do a different set of actions. And so this is I want to see how these two mechanisms might interact with mitochondrial function. So the way we did this was to take an insulin receptor Flux mouse, and we treated it with an adenovirus containing CRE for 2 weeks. So this is a relatively acute specific takeout of the hepatic insulin receptor. This mouse was made by Ron Kahn. It's been studied a lot. So it's a little bit new. We're digging into the pathways a little more here. So what happens is what you would expect, that you get an increase in gluconeogenesis when you lose insulin action at the top. That's expected. What you also find is that you get this induction of all the oxidative pathways that might be required to drive that gluconeogenesis. So you get an increase in the TCA cycle, an increase in ketogenesis. And there's not much data about what happens to oxidative stress in these mice, but if you read the paper, one of Ron Kahn's first papers on this mouse, what he says is that after about a year, the mice die of liver failure, and he thought it was because of oxidative stress. There's not much data, but it might be interesting to look at these mice after a longer term as well. So that leads me to how that might happen. So we tell our biochemistry students that gluconeogenesis is energetically costly. It's expensive. What do we mean by that? What we really mean is that it takes a lot of ATP to make one glucose molecule from pyruvate. And so in fact, people have tried to quantify this. It takes about 40% of the liver's oxygen consumption, which is, by the way, about 30% of your entire oxygen consumption for your body, just to supply gluconeogenesis during fasting. So that's a huge load, an energetic load on the liver. But what that means is that if you progress to a state where you have elevated gluconeogenesis, like in diabetes, that you've increased the burden on the liver. You must, so this isn't, my opinion, this is just math. It's stoichiometry. You must increase oxygen consumption by 25% to support this increase in gluconeogenesis. So what we thought we would try is to limit the liver's ability to do that. So let's take a liver, make it fat, make it insulin resistant, but then take away its ability to induce gluconeogenesis. And we did that by knocking down Pepsi K by 70%. We've worked a lot in this Flux mouse. We know a lot about it. We were able to take it down 70%. And when you do that, this is what happens. It doesn't impair the mouse's ability to produce glucose in the basal state. But what it does is that when you put them on a high-fat diet, the normal mouse gets this induction of gluconeogenesis that we all appreciate, but the knockdown mouse is prevented from doing that. This is what happens to oxidative flux when you calculate it using all of the tracer approaches that we use. The normal mouse gets this induction of oxidative flux, presumably to support that, while the knockdown mouse, it's prevented. So this allows us to test whether this induction of oxidative metabolism is in any way related to the oxidative stress and inflammation that we see. In fact, these mice don't have differences in fatty liver. They have just as much fat in their liver as the wild-type mouse. So it's not fat, it's actually the pathways that we've impinged upon. And when you do that and you study the reactive oxygen species using a dye like dihydroethidium, what you can see is you put a normal mouse on a high-fat diet and it just lights up. Lots of oxidative stress occurs. But when you knock down the liver's ability to respond or to induce these pathways metabolically, they're protected. Not as much reactive oxygen species. And so we think that it is this protection that leads to this protection, a protection from inflammation, a decrease in markers of inflammation, including IL-6 and TNF-alpha, and also the actual signaling pathways through the NF-kappa B. The mouse is protected from all of those, but it has just as much of a fatty liver as a normal mouse, but we've limited its ability to induce all of these other pathways. So it leads us to think that perhaps by suppressing this oxidative metabolism, we limit the oxidative stress, reduce the inflammation, and decrease the feedback on insulin resistance. In fact, the mice are more insulin sensitive when we do this. I didn't show that data. And then overall prevent the liver disease. And I wish I could stop here. I wish I could say that's it, no more. But that's not the whole part of the insulin signaling pathway that I mentioned, right? This mTOR pathway is part of a pathway called, or part of a pathway that we consider as an insulin signaling paradox. And the reason we call it an insulin signaling paradox is because when you have true insulin resistance in the liver, of course you will decrease your ability to signal in this pathway. But for reasons that we don't understand, you don't decrease your ability to signal to pathways along the mTOR pathway. So we wanted to see how this might impact the situation. In fact, when you put a mouse on a high-fat diet, this is mTOR activation measured by S6 phosphorylation, you get an induction in S6 phosphorylation. Even though it's downstream of the insulin receptor, it's still active. So we did the same kind of experiment that we did with the insulin receptor knockout. We took a mouse that was Flux for this kinase that's the mTOR activator, and it impairs. It inhibits mTOR, and we knocked it out. So then what you have is a constitutive activation of mTOR in the liver. The same conditions, about 2 weeks of activation of mTOR. This is constitutive mTOR activation. And what do you find? The exact opposite. So when insulin signals through the mTOR pathway, you impair oxidative metabolism. You decrease TCA cycle. You decrease ketogenesis. So this must be happening at the same time that the insulin receptor is driving things forward, mTOR is driving things backwards. So going back to that one of the early slides that I showed you, that a high-fat diet in mice initially causes this loss of flexibility in this pathway, the loss of the ability to go from fed to fasting. You find yourself in an intermediate state. And what does mTOR do? It does the exact same thing. When you constitutively activate it, these mice, even on a chow diet, are just locked in this intermediate state, somewhere in between fed and fasting. So mTOR activation in response to insulin must be important for regulating this nutritional flexibility. So how does that happen? We're not really sure. So some of the initial work by Sabatini certainly showed that mTOR is important for regulating HMG-CoA synthase through PPAR-alpha. We found that as well. We also found that acetyl-CoA carboxylase was induced in these mice, so it could be changes in malonyl-CoA concentration. But one of the more fascinating things that we found in this mouse, and is known to some extent in the literature, is that it's really important for fasting-induced autophagy. So this is what happens when you do the liver size of a mouse when you fast it for 24 hours. It dramatically decreases. This has actually surprised me. I guess it's not unknown to the field, but it surprised me personally that you fast a mouse for 24 hours and they lose 40% of their liver mass. It's autophagy. They're consuming the amino acids and the glycogen that's in the liver. When you do that experiment in a mouse that has mTOR constitutively activated, it can't do that. The livers start big in the first place. They have hepatomegaly. So they have these big livers, and they don't respond to fasting. They stay big. So one possibility is that they're just keeping all of those reserves, and they're not allowing the mitochondria access to those substrates. So it's almost like an internal substrate cutting off the internal substrate. But this is where we're going now. And so mTOR activation increases liver size. Insulin receptor knockout decreases liver size. It's more catabolic. But true garden variety insulin resistance is both. It's a paradox. And so what we've done now is made the double knockout. So we tried to recapitulate garden variety insulin resistance molecularly by taking out the insulin receptor but activating mTOR. And what you get is a recovery of liver size. And we don't know yet what happens to the fluxes and what happens to oxidative metabolism, what happens to reactive oxygen species. That's where we're going. So just to summarize, when you lose insulin action, you induce the anabolic pathways that require oxidative metabolism. We think this contributes. I'm not trying to say this is the whole story, but it contributes to oxidative stress. And it's probably okay under the initial setting. But mTOR also has a role to play. And what mTOR does is it almost opposes that. Maybe it's protective because it's stopping this hyperactivity. But over time, maybe it loses its ability to do that. It does other things that I haven't even talked about. mTOR is required for de novo lipogenesis during insulin resistance. So one of the things that we also pay a close attention to is lipid synthesis as a contributor to the lipid in the liver. It induces that. It also, and these things contribute, of course, to the metabolic syndrome. It also may play a role by making the liver bigger. So now you have hepatomegaly, a more active liver, and that could lead to lobular hypoxia. These are things that we're trying to study. And finally, if you have too much oxidative stress, you get inflammation and the liver disease that I showed in the beginning. So I'll stop there. This is a picture of the lab. I need to thank Jeff Browning and Elizabeth Parks, who did most of the human studies, and Blanka Kusajova, who did most of the signaling studies that I showed you. And of course, we're very grateful for funding, and I'm grateful to have you here listening to my talk. Thank you.